A generic drug proves one thing: the same active ingredient at the same exposure in the body. Two studies, an argument is made. A biosimilar can never make that claim. The molecule is grown in living cells and no two batches are truly identical. So what does FDA accept instead? That answer has changed twice in the last year. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to build the analytical packages behind biosimilar and generic filings. Today, we are going to walk through what a 351(k) application actually has to prove and how that bar has shifted since October 2025. Before we start, I would request you one thing. If you have found anything valuable from this channel, please like and subscribe this channel. This gesture of appreciation really means a lot to us. Now, let's start with why the word generic doesn't fit. Generics comes from the Hatch-Waxman Act of 1984, and that pathway rests on one idea: the same active ingredient made by a different company reaching the body at the same exposure. Biologics had no abbreviated rule at all until the BPCI Act in 2010, which created a section 351(k). But the Standard Congress wrote there is not the same, it is highly similar. That one change in wording sets up every study a biosimilar sponsor runs. Now, what highly similar means in law? Section 351(I) says two things. The product must be highly similar to the reference product apart from minor differences in clinically inactive components. And there must be no clinically meaningful differences in safety, purity, and potency. Read that closely because it's doing a lot of work. Differences are allowed. What is not allowed is a difference that shows up in the clinic. The job of filing is not to prove the molecules are identical, it is to show that where they differ, it does not reach the patient. Put the two applications side by side and the gap is easy to see. An ANDA rests on chemical identity plus a bioequivalence study, usually pharmacokinetics in healthy volunteers with the confidence interval of area under curve and Cmax inside 80 to 125%. A 351(k) BLA has no short version of that. It carries a full comparative analytical assessment, its own drug substance and drug product CMC package, a human PK similarity study, and an immunogenicity assessment. Same commercial goal, a cheaper version of a proven medicine, a completely different evidence base. Now, why can't a biosimilar simply show identity? There are three reasons, and all of them are physical. First, scale. A small molecule holds a few dozen atoms. A monoclonal antibody holds more than 20,000 folded into a shape that carries the activity. Second, the process. The protein is expressed in living cells. So what you make is a population of closely related forms, not a single species. Third, structure drives effect. Shift the glycan profile and you can shift how the antibody recruits immune cells. Now, let's talk about totality of evidence. FDA calls its approach the totality of evidence, and a shape of that stake matters. The base is the comparative analytical assessment, attribute by attribute, method by method against many lots of the reference product. Above it sit human PK similarity and immunogenicity. At the top sits the comparative efficacy study, and this is where FDA's thinking has changed. In the October 25 draft guidance, the agency said an efficacy study is no longer the default because a well-built analytical assessment is generally more sensitive at picking up real differences than a clinical endpoint is. Now, where does the analytical work really get hard? There are again three places. Come up again and again. Glycosylation, because fucosylation changes ADCC activity, which is why the glycan map is compared peak by peak. Charge and size variance, because aggregates and acidic species feed immunogenicity risk, so SEC, CEX and orthogonal methods all have to agree. And potency, where one assay is never enough. Target binding, Fc receptor binding and a cell-based readout each cover a different gap. At Resolve Mass, we have supported sponsors whose whole comparability story turned on a single unexpected shift in one of these attributes. Now, put in order. The program looks like this. You source and map lots of US licensed reference product, so you know its real range rather than one batch. You set the quality target product profile from that range. You tier attributes by criticality, then match each tier to method sensitive enough to see the difference. Clinical PK and immunogenicity come next. And the 351K filing after that. One more thing worth knowing. The March 2026 draft Q&A revision also opened doors to non-US licensed comparators in the same PK studies where it can be justified. Now let's talk about key takeaways from this video. A generic proves sameness, a biosimilar proves highly similar with no clinical meaningful difference. The analytical package carries the argument, not the clinical trial. The 2025-2026 drafts moved that work earlier into the lab, and the sooner you know your reference product's real range, the simpler everything downstream becomes. If you are building a 351K program and you want a CDMO CRO partner in Canada who treats comparative analytical work as the core of the filing, Resolve Mass Laboratory supports pharma and biotech teams from reference product characterization all the way through submission to the. Before you go, I want you to do a few things. Number one, subscribe this channel right now. The more you subscribe, the more valuable pharma insights and expert tips and industry updates we can bring to you. Also, please comment your feedback. This will really help us to improve the content, quality of the videos, and many things. And finally, share this video to at least one person who you think this would really help them. Thank you so much, and I will see you in the next time. Until then, goodbye for now.